see everything that's going wrong with the world and those who lead it. We just feel like we don't have the means to rise above and beat it. So we keep waiting, waiting on the world to change. We keep on waiting on the world to change. It's hard to beat the system when we're standing at a distance. So we keep waiting on the world to change.
This is Dr. Ed Park. I'm recording part 4 of a 9-part series on exosomes. This lecture is entitled, What Role Do Exosomes Play in Cancer? If we ask the question, what is cancer, it's reminiscent of the Supreme Court Justice who wanted to define pornography. He said, I can't tell you what it is, but I know it when I see it. And unfortunately, that's the case with cancer as well. It's overly broad, it's impossible to define except post hoc. And in fact, the experts in this, the pathologists, go by things like appearance, how active the nuclei are, how active the organelles are. A slide down at the bottom right shows just what they're using to base their assumptions and conclusions upon. Now, the behavior of cancer includes different surface cell antigens and different behaviors in terms of being inhibited by contact with other cells. Molecularly speaking, they probably send and receive different messages via the exosomes. So there's a shift in thinking towards a more ecological model of cancer. Finally, cancer stem cells, which is one of the best hypotheses of what cancer really is, it teaches us that stem cells are already telomerase positive and immortal, and they may be where cancer is taking place, not in regular run-of-the-mill differentiated cells. So it's of particular concern when a Clark Kent superhero cell like that loses the ability to kill itself. That might look like senescence, but it also might look like cancer if it has turned off its program of cell suicide by losing one or more of the genes involved with that process. When we look at commonly held beliefs about cancer, there are 3 that are considered true but are actually myths or delusions. One, that it's rare, second, that it's hereditary, and the third, that it's deadly. In fact, cancer, if you look at autopsies of young men who die in motorcycle accidents, can be found in their prostate. And in fact, one of the subjects of the blogs I did and videos was the Oncoblot test, which came onto the market and left the market because it was detecting cancer at such an early stage that in many cases it would never have manifested clinically. If you look at the comments on the posting, some people were mad because they got prophylactic mastectomies or other organs removed. And in fact, in real life, a lot of the cancer that we get, perhaps millions of times over, is never actually going to manifest with a healthy immune system. Number 2, very little of cancer is attributable to hereditary or genetic factors. Of course, the BRCA mutation is the exception that proves the rule, but by and large, most people get cancer just from being alive long enough to acquire the genetic damage in those stem cells. Finally, the idea that cancer is deadly is pervasive, and yet, if we talk about hotel fires, the MGM Grand Hotel is the exception that proves the rule. Usually a hotel fire gets put out in the ashtray, or on the stovetop, or in the plug with water or fire extinguisher. There are also barriers that come down, sprinkler systems. So if you talk about a hotel fire, in general, most fires in hotels, 99.99%, do not result in an overwhelming destruction of the hotel. Let's look at my cancer pyramid from my first book, Telomere Time Bombs, Diffusing the Terror of Aging. What we see is that cancer for us is the tree above, treated with surgery if it's local, radiation if it's spread to lymph nodes or regionally, and then chemotherapy if we want to poison cells all over the body. Of course, that comes with a highly global and destructive damage consisting of brain fog, from neurological damage, loss of the immune system or anemia, destruction of the gastrointestinal system, and also hair loss. These are all rapidly producing cells that are poisoned in their stem cell versions. What's ignored is the pyramid below, of course. Before we manifest any clinical cancer, we have your own immune system. Cells are constantly being watched by your adaptive immune system, consisting of natural killers and cytotoxic T cells. And they're also displaying a signal to cells to murder them when they're hijacked by viruses or by cancer. Below that is the P53, the cell suicide watchman of the DNA, and every cell is endowed with the ability to kill itself, repair itself, or stop reproducing. So this is also a very important mechanism that prevents many of the cells from becoming clinically evident cancer. And finally, in my model, telomere erosion, which is causing a lot of the mutations and the cancer, is the first line of defense. So if we have high telomerase activity, we have slower rates of mutation, slower rates of depletion of the immune system, and this will help to prevent cancer. The question of when is cancer is a pretty obvious one. Cancer is usually associated with aging. It's usually that way because they're coincident and they're causal. As the stem cells age, their telomeres shorten and they acquire mutation. In fact, aging also allows for mutation and depletion of the immune system and accumulation of genetic defects by this replicative senescence that we talked about. If we look at the article by Willett et al. In JAMA, or the Journal of the American Medical Association in 2010, they had a very interesting study showing that the shorter the telomeres, the higher the incidence and lethality of cancer. This histogram on the top shows exactly that. In rare cases, when you do have the BRCA, for example, or other cell cycle or apoptosis type mechanisms, even genetic repair, if you only have one good copy from your parent, then that cell is vulnerable. And when it loses the only remaining good copy, you do get this increased rate of cancer formation. And that's known as loss of heterozygosity. Or you only had one good copy, like a kidney. You had a 50% gene dose from the get-go, and then losing the other puts you in failure. Some critical mechanism of cell surveillance was destroyed, and then you're off to the races. We have to understand that after illness, stress, and even cancer chemotherapy, the risk of cancer also increases. So our interventions are not neutral by any means. One can view cancer as a failure of apoptosis, as I've suggested in the previous slides. We know from elephants that rarely get cancer. We assume it's because their genome has multiple copies of P53, or this watchman. If we look again back at the pyramid I created, apoptosis is a natural function of cells. They kill themselves when they're genetically damaged. And that probably is a much ignored aspect of how we age, get old cells, and accrue cancer. There are other tumor suppressor genes involved with cell cycle regulation that have been proven to cause an increased incidence of cancer. But if you drive any car for long enough, the car parts will deteriorate. So we really must view cancer as a very common and inevitable, if you will, result of being alive for long enough. Now, there are other methods to destroy cancer before it becomes clinically evident, and those include telomere attrition itself. The fact that certain cells are not telomerase active and that they are disposable, if you will, like paper cups or leaves, means that they are not going to be able to endow their daughters and their progeny with immortality. Secondly, I can't stress enough the importance of immune surveillance. The system is designed to detect and destroy cancer. And then finally, contact inhibition. And we learned this from the naked mole rat, who has very long life and who also does not have the same kind of problems with contact inhibition loss. In other words, the cells appear to play very nicely with each other and don't invade. So our hope is that exosomes from a positive ecology of good cells can inhibit apoptosis, or in the case where it's needed, promote it and encourage the destruction of damaged cells, both by rejuvenation and optimization of the immune system and by introducing much needed and deleted proteins. Revisiting cancer as failed immunity, we see that cytotoxic T cells and antigen presenting cells will work together to kill cancer. In the case of Jimmy Carter, he took a PD, that's programmed death ligand 1 inhibitor called Keytruda, an antibody that just blocks the T cell inhibition, and as a result, his T cells were able to recognize and kill the cancer. Other immune therapy modalities include flooding the patient with their own cytotoxic cells and clearing cancer. Now, it's interesting that the immune side effects or the autoimmune side effects of Keytruda point to the fact that PDL1 may be there for a reason. It may be there to help us grow old by allowing cells to live with damage. When you introduce Keytruda, the cells become aggressive and they kill the cancer, but they can become overly aggressive, and we know that there's a higher incidence of clinical autoimmunity. So this points to the fact that autoimmunity, or the ability to destroy one's own cells, is also a mixed blessing. It can help to clear cancer, but it can also help to kill innocent bystanders or semi-innocent bystanders. So they may be opposing missions. Although, if your system is optimized and working well, it should be able to do both. It should be able to walk and chew gum at the same time. We can also view cancer as cell ecology. Now, exosomes are a gateway to better understanding, and a lot of the early research in the last 10 years has been about how exosomes from cancer ecologies differ. In the last few sentences of my first book, I hypothesized that we'd get somewhere when we started understanding stem cells as if they were social insects. And in fact, that is a theory that was come about or posited 130 years ago by a man named Stephen Paget. He said that seeds and soil were the important thing. In other words, that cancer is not just about the seeds, but the soil has to be prepared. And in fact, this is being pointed to by some of the exosome research. If you take exosomes from somebody who is succumbing to cancer, they will promote transformation in an otherwise healthy person. And likewise, if you take exosomes from someone who has cured their cancer, they may have the secrets to making the soil less amenable to being planted. So we see that bad ecology is both the cause and the emergent property of aging and of this increased entropy resulting in cellular chimerism, or mixed sort of behaviors. And cells all become strangers to each other, and you throw on top of that gene mutation, gene silencing by methylation, and other interfering RNAs. And imagine if you had an interfering RNA for p53, then that would be deleterious or harmful to cell cycle control and suicide. Much of the research going forward and the investment will be as a mode for drug delivery, because it's this perfect time-release capsule that goes right to the cells and is protected from degradation by proteases and other things in the blood. Now, the goals are intellectual property driven. People want to make money, patent something and sell it, not necessarily outcome driven. So if we're more interested in the general takeaway lessons of what a good exosome profile would be from a patient that's surviving and winning versus one that's succumbing and dying, then perhaps research on this level is a bit behind by nature of what drives people to invest time and money. We have to understand that traditional chemotherapy is mutagenic in most cases and causes a lot of excessive collateral damage. But in the future, I guess the hope would be that we'll have personally tailored solutions that would enhance the mRNA copies that are missing. If you do a test on a cancer and you find that in this particular clonal variant, the p53 or the p16 is gone, then perhaps you can introduce exosomes that have p53 and p16 messenger RNA, giving them that temporary ability to make these proteins and undergo suicide. In addition, we can also do something to silence bad genes with microinterfering RNA. For example, the FOXO4 protein is a senescence promoting protein that we make, allowing the cells to survive in a damaged senescent state. Of course, if we had an miRNA to block that transcription, then we might have an increased efficiency of senolysis or old cell death. Finally, contact inhibition via FAK is hugely important in terms of cancer metastasis, and an interfering RNA would be helpful there. Anti-angiogenesis factors have been experimented with, but the results are mixed. So vascular endothelial growth factor, something to inhibit that, might provide much needed respite from the nourishing and the growing of cancer at its distant sites. In conclusion, since 2017, very few doctors have been using exosomes from mesenchymal stem cell sources. Some also use them from amniotic, which is a very heterogeneous population, and from cord blood. Now, none of them have reported increased cancer rates, but this is a theoretical concern. In the future, exosomes from other healthy cell types, such as nerve, cartilage, kidney, might provide organ-specific, promising treatments for regenerating those tissues. Now, orthopedic indications are very theoretically sound because mesenchymal stem cells are what are recruited to regenerate damaged joints, cartilage, muscles, tendons. And so I got into this because some of the MDs using exosomes were reporting very promising anecdotes. This leads us to the fifth lecture in the series, the history of regenerative joint therapy. So I hope you'll tune into that to learn more about the role of exosomes in musculoskeletal regeneration. Thank you.